So we're taking viral nucleic acid and we're hurling them all into a sequencing machine. Our main purpose is to identify new human pathogens, animal pathogens, and also to add to the greater understanding of the evolution of viruses. This is the discovery curve for human viruses over the last century. And this sort of research, literature search stops just when basically high-throughput sequencing started. Since then, there's been a great peak in the numbers of human viruses that have been discovered. I should also point out that the Bay Area is very strong in viral discovery. There are several groups involved in this. There's Novartis, there's the laboratory of Joe DeRisi and Charles Chu, both at UCSF. So where are we looking, what kind of disease are we looking at to identify new pathogens? Gastroenteritis, diarrhea, there's still a large fraction of unexplained cases, even in the US, but especially abroad. Respiratory illnesses, a large fraction remain unexplained. Encephalitis, a large fraction of unexplained cases. And of course, the big targets are autoimmune diseases and cancer. Now, in order to not have to sequence too deep and not resequence the human genome, what you want to do is to purify viral particles so that your sequencing is focused on their nucleic acids. And the way we do that is basically very simple. We just filter homogenized tissues or fecal supernatants or plasma. And then we take the flow-through after the filtering, which will retain basically bacterial cells, eukaryotic cells, and let the viruses through. We will remove the free-floating nucleic acid by throwing a cocktail of nuclease, DNAs and RNAs. Most of those biological fluids are just loaded with human DNA as well as RNA. And you want to remove them as much as you can. And then we use what's called a random RT-PCR. Basically, the primers are completely randomized at the 3' end, so that theoretically they will bind all along an RNA or DNA stretch, even reverse transcriptase, which is also a DNA polymerase. They will extend the first strands. We will then denature that cDNA, and we will add the same primer to make the antisense. And then we will take that double-stranded cDNA, and we will add just that part of the primer, and we'll go through 30 or 40 rounds of PCR, enough to see on a agarose gel. Now, the process at this point is we throw it all into a sequencer. We went from 454 to Illumina recently. You collect the data. You can, those primers, you can use different types of completely arbitrary sequence, and you can use 50 different ones. So you can have 50 different tags. You can pull the PCR products into one run. Computationally, you can deconvolute so that you know the reads, the origin of each of the reads into your sequencing run. And then what we do is we take the sequencing read, we remove the primers and so forth, and then we say, can we assemble contigs to larger sequences? And if the viral titer is high enough, you will basically resequence the full genome right off the bat. More often than not, in biological fluids, depending on when you get them during the infection, you'll have more or less reads. But if you have nice samples, you can basically resequence the whole genome. This is a picornavirus. So we take then the contigs and the singlets, and then we basically put them all through a blast search, where we're looking for protein-level homology to already sequenced viruses. And then we have a nice website where we can peruse the data and look at a lot of alignments. So we know the technology is fairly good because we've basically rediscovered a lot of viruses. And more importantly, we've gotten viral genomes or fractions of genomes from all the different types of viruses, the DNA, the RNA, the double-stranded, single-stranded, circular, fragmented, multiple fragments, and so forth. So it's a very non-specific approach. Here's what sort of raw data looks like. If this is a fairly obvious to recognize virus, you can look at the alignment. You can look at the E-score, which is highly significant. And you know right off the bat that you have a new virus. Here's a more iffy case where the E-score is more borderline. You have, you could argue whether this is a background or not, even based on the E-score. But when you further extend these initial hits, typically, you will get better and better E-scores, expectation scores, so that you will know then at this point that you have a virus on your hands. This was a new Callice virus. So this is from my bioinformatic help guy. The perception of bioinformatics, it can do wonderful things really quickly, perfectly. But the reality is more like this, where there's a lot of rewriting codes that's necessary all the time. But it is essential to have good bioinformatic backup, as well as fairly good computing power. So in effect, what we're doing, we're looking for new viruses that are related to known viruses. So if this is like a viral family that we already have many taxons, virus species already sequenced, we can recognize those that are fairly closely related. They're going to have fairly good E-scores, that is low E-scores. As the viruses are more and more divergent, the E-scores will drop. And at one point, we may just have viral genomes, but we won't even recognize them because they're going to be so far from what is already in the database that is annotated as viral in GenBank. So as an example, a virus on the edge of recognizability, you will pick it up, you will sequence it, and you'll add it to the database. Now, at this point, what happens is you're now able to see more sequence space. You'll be able to recognize viruses that were previously unrecognizable because now they're closely related to your new genome. So just adding data to the database, a viral database, has some utility. Now, of course, there remains a lot of sequences which do not blast to anything. And they're going to be bacterial, fungal, but some of them may very well be viruses. And there's many ways to start looking at this sort of dark matter, maybe new viral families, and some of them might be involving trying to do virtual protein folding. If you frequently detect the same long contig that doesn't look like anything in the same kind of symptoms, then you may sort of postulate that it's a new virus. You could also try nucleotide composition. Viruses, at least certain viral group, have different nucleotide compositions. But when you apply this technique now, here's what you get, for example, if you have a clean viral prep. This is a live attenuated herpes vaccine, which I highly recommend for shingles. And when you sequence it, you can basically resequence the entire genome. Now, notice those peaks, which really tell us that this random RT-PCR is not so random, but random enough that you get fairly good coverage over the whole genome. You can also look at quasispecies, that is a very complex RNA viral mixtures. For example, it is a polio vaccine with the three genotypes, and you can basically resequence them all. And you can also look at some depth at particular position where for example, some reversion mutations to pathogenic polio occur in certain regions, and you can look, is there a low fraction of those revertant in the vaccine? And the answer is no, of course, because we all know that this reversion takes place during replication in the gut. And every once in a while, what you can see is something unexpected. This is a rotavirus vaccine where we found basically a contaminant from a pig virus. Luckily, this pig virus is extremely common in the food supply. Whenever you eat pork, you're probably going to eat some of that virus. And as far as we know, it does not even infect humans. And the origin of that contaminant may have been porcine trypsin that is used to actually passage rotavirus. But the FDA decided to sort of tolerate this because it's such a good vaccine, while the company slowly gets it out. Now, the other thing you can look at is more complex material. Here, for example, we've been looking at a lot of fecal samples. I kind of like that ad because we do a lot of that. And so we looked at, for example, pigs on a high-density farms. What kind of virus are they shedding? And what we realized is there's a huge amount and a huge diversity of viral shedding, mostly picorna, parvoviruses, coronaviruses. Whether you're looking at pigs, little piglets with diarrhea, or healthy pigs, they're both shedding a lot. And it's quite likely that this by and large, very little consequence because of maternal antibodies. As a matter of fact, farmers will tell you if your little piglet doesn't get to suckle, it'll probably die within weeks from some nasty infection. You also get, when you look at fecal material, a lot of stuff from plants, which is from the diet. So for example, these are plant viruses that we found in human stool people with diarrhea, tourists with diarrhea in Nepal, and you can get a fairly good idea of their diet. Now, here are stool samples from kids with a non-polio acute flaccid paralysis from Pakistan. This is the first set of samples we looked at where we found a lot of new viruses. And I think the reason is people just hadn't applied this sort of very simple technology to these sort of samples from developing country. And so a lot of co-infection in those sick kids. And this fatal case here, this kid had actually 5 mammalian viruses and of course, a little plant virus as well. Now, a lot of these viruses are asymptomatic. As we saw with the pig, the little piglets, you can shed a lot of viruses and without consequence. These are 2 kids sampled every week for the first year of their life. They were never hospitalized. They're just regular kids. And what we did is not deep sequencing, but PCR for a whole bunch of human enteric viruses. And what that shows is just the amount of infections and co-infections going on in healthy kids during the first year of life. Some of them are quite long, and some of them are really just here for a week or so. So a lot of asymptomatic infections. The exact virus doesn't really matter, I don't think. Now, so when you're applying this deep sequencing for viral discovery, the most important thing is the sample. If you have a high titer, you don't actually need deep sequencing. You can just do this sort of random RT-PCR. You can subclone into a plasmid. And if your sample is a high titer, here we had a titer of about 1 million copies per mil from a plasma from an injection drug user who had all the symptoms of acute HIV infection, but he was HIV RNA negative. But he was pretty hot with this new virus, and here is the assembly. So the point is, it's all about the sample, the biological sample. When we sequenced and we put it together the genome, we had a new human parvovirus, which we called parvovirus 4. A point to be made is that there are also microarrays that have been developed for viral discovery or for diagnostic. These are very good, and you can put hundreds of thousands of probes on them. But a virus that is too divergent to what is already known will not stick to some of those microarrays. So maybe a more sensitive way to detect these highly divergent viruses is to look for protein-level homology. So a little more about the parvoviruses. They're the smallest known viruses in size, as well as, well, they are smaller in genome size. They're very tough viruses. They resist, they can be sitting in dirt and be infectious weeks later. They're very hard to inactivate, so they're a problem for plasma industries. Now, when we studied that, the known human parvoviruses was B19, which is generally a harmless infection in most kids, but can cause some anemia-like symptoms in certain susceptible population. And then the adeno-associated viruses, which as far as people know, doesn't cause any symptoms. What we found was parvo 4, and also these other parvoviruses, which I will not get into. I'm just going to focus on that one. So it has a very deep branch. It's quite divergent from the other one, and it's the 3rd group of parvoviruses. Well, actually, there's a 4th now infecting human. Now, of course, when you have a new taxa, actually this is a new genus in this family, people design now primers to it, and they go fishing for relatives. And what has been since seen is 3 genotypes of this human parvovirus in plasma, and also it's been found in primates and farm animal, and now there's even a bat one. Now, the next step is to develop serological reagents to express these capsid viral-like particles in an insect cell system. And then you can look for what is the epidemiology of this infection. And turned out that the people with the antibodies to parvo 4 are mostly individuals who have blood contacts, that is, injection drug users, or hemophiliacs who are treated with coagulation factors, and a small percentage of men who have sex with men. But it's not really found at any significant level in heterosexuals. The other important thing about this virus is it's even the heat treatment that plasma products undergo for safety purposes do not inactivate this virus. And this is shown here by seroconversions in hemophiliacs who were treating themselves with clotting factors. So is it a pathogen? Well, at this point, it's still not clear. There are anecdotal detection of these viruses in cases of encephalitis from David Wong and his collaborators. Also cases of powerful detection in case of fetal hydrop, which is also a disease that B19, the other parvovirus, can cause. When you look at prevalence by PCR, what you found at very low level, it's a bit like B19, which is present for life in tissues at very low level. Here we find it in plasma, in about 2% of blood donors from LA. When we looked at those more individuals with those symptoms of acute HIV infection, we found a significantly greater fraction of them were positive. But that doesn't, because it's a chronic infection, it does not mean that the symptoms were caused by this parvo4. It may just reflect the sort of the higher exposure in general of this population. So we're still not clear whether this is a human pathogen or not, but it's certainly quite common, and it may have sort of hitchhiked its way along with the HIV and the Hep C epidemic to become now quite prevalent throughout the world. Quickly, I'm going to go through some picornaviruses, which is the polio is the prototype. And when we started that, there were only really 3 genera of picornaviruses known to infect man: Par-Eco, which causes a lot of encephalitis, hepatitis A, which causes hepatitis, and then the enteroviruses, which are an extremely complex, large group of viruses causing, which also include now the rhinoviruses, so they cause enteric problems, respiratory, and polio. And then the cobuviruses, Aichi viruses, which causes diarrheal problems. Since we started that, there's been quite a few additions. The cardiovirus group was shown to infect humans. This was found by several groups. The cozoviruses we found in kids in Pakistan. It's an extremely diverse group of viruses with now 5 species, at least 30 genotypes. So it's starting to resemble the enteroviruses. And the salliviruses, also called classiviruses, was found almost simultaneously by 3 groups. And that's been associated with diarrhea. Now we look at the prevalence of nucleic acid in feces of these cozoviruses, and we compared it to the enteroviruses using fairly generic primers for this diverse group of viruses. We found these enteroviruses had incredibly high fraction of feces from kids, both with flaccid paralysis and without. So it's just sort of common infection. And cozoviruses were found in nearly half of all the samples we looked at. So they're extremely common infection, and it's surprising to me that it took that long to actually find these viruses. Now, the point to be made is there's such diverse groups of viruses, and like many infections, you probably have only a few genotypes within a much greater sort of a zoo of diversity that are actually pathogenic. And also remember that these things can change quite rapidly over time. But it does complicate disease association studies for this sort of diverse viral family. Very quickly, another RNA viruses. When we started off, there was basically one group up here of astroviruses causing diarrhea. David Wong and his group, who used to be here at UCSF, found a second group. And then we and also David found a third group. So, just a few years ago, there was basically one group of astro, now there's 3. And same for humans, for animals. There used to be just one pig astroviruses. There are now, I've stopped counting, 3 or 4. Same for other viruses. Now, people interpret this sort of intermingling of species along the phylogenetic tree as reflecting probably quite frequent transfer of these astroviruses from species to species. Otherwise, all the humans would be on one clade. We've also been looking at animal samples with a great collaboration with Pat Pisavento in UC Davis. She has a great eye for identifying interesting samples. This was dogs with vasculitis and a problem with their kidneys. And here's the picture of a normal and affected kidney. We found 2 viruses in those tissues. One was a new circovirus, and before that, infecting mammals, there was only this pig circoviruses, which I mentioned was, one of them was contaminating the vaccine. PCV2 is actually the pathogenic form, which is a very common pig virus and a big problem in the pig industry. So we found the canine circoviruses. All the other circoviruses actually infect birds. And we also found, and we'll talk about this whole sort of sister clade called cycloviruses that is, may also be infecting mammals. Well, it is infecting mammals because we find it in tissues. It may also be infecting man. And then another bocavirus in this tissue. So what do you do when you have such a new discovery? Now it's in the tissue, it's not in the feces. So we're pretty sure it's actually infecting these dogs. Pat went and did some in situ hybridization with the genome of these circoviruses, and she finds very hot cells in the macrophage, but also in the around the vessels with vasculitis. She developed antibodies to that, and again, she showed expression of this circovirus protein inside macrophage. And also, looking down now with the EM, you could see nice arrays, paracrystalline arrays of possibly inclusion bodies of the virus. So putting the virus in the right place, I think, is one important step towards showing that it might be pathogenic. So we now have many new viruses being sequenced using metagenomics. Many are orphan viruses where we actually don't quite know what they're doing, if anything. It may be that they're pathogenic in some circumstances, in some genetic background, with other co-infections, in seronegatives only. But this is going to be quite complicated to tease apart. And some we can more confidently associate with disease. I mentioned this complexity of the diversity of these viral clades. There's phylogenetic evidence of frequent cross-species jump, this mingling of species across the tree of the viruses, and a high rate of co-infection in both healthy and sick young human and animals. Quickly, there's a list of the new orphan viruses in some of these groups. A lot of new parvoviruses have been identified recently. A few years ago, it was really basically only B19, and now there's 3 other groups. Picornaviruses, we mentioned the COSA, the SALI, causing maybe diarrhea in the cardio, still unclear what they're doing, but probably diarrhea as well. And astroviruses, and I didn't mention at all, but the polyoma, and we haven't really anything to do with this, but there used to be just a few polyoma a few years ago. They've like tripled in numbers in the last few years. This is the number of viral family known to infect man, and there is 20 or so, 22 families, and some of them have greatly increased in the number of human viruses now known, and including the polyoma here. And some more even maybe we'll be able to add new families, such as the cycloviruses. We're finding lots of circular single-stranded DNA genomes in different types of samples, not always sure whether they're actually infecting the host in which you find them, but I think in time, with the right sample, the right epidemiology, we'll know a lot more about what are the hosts and what disease they cause. So what's next for the orphan viruses? There's a need for a lot of molecular epidemiology, basically case control studies, comparing the virus load and prevalence in symptomatic versus matched healthy control. That's very important to, in those case control studies, to actually very closely match the two populations. You can't compare kids to adults, Americans to Pakistanis, and so forth. Showing seroconversion is during convalescence, a post-infection. Also shows in time that the virus was there, and had not been there, for 10 years before as a chronic infection. It's good to show RNA and protein expression at the site of pathology to put your virus, in the right, next to the body, so to speak. And of course, animal models is one way to find out more whether there's at least pathogenic potential for some of those viruses. So I'd like to acknowledge, too bad we can't read that, people who did the work, past members of the lab, and of course, my great collaborators. Everybody is really essential, and of course, NIH and BSRI funding. Thank you.
Two related questions. First of all, what fraction of the viral genome do you see before you call something a virus? And second, do you see any evidence of recombination if you have multiple viruses or evidence for multiple viruses at the same
So
time?
recombination is quite common within a clade, depending on the type of viruses. Picorna recombine very frequently. We are now finding, as we pay attention to more of those odd viruses that are harder to detect, that some of their, you can make trees that are discordant depending on the genes. So they do look like they're a bit of a hybrids of different types. We have, for example, viruses that look a bit like a partly parvo, partly circo. So we do seem to, as we grow the taxonomy, if you want, we're finding not only new species within the Picorna, but new families as well. So new deep clades with deep branches. In the single-stranded circular DNA family, we may have some they may be recombinant, but they're so old that the evidence is still sort of, I think, borderline. But yes, recombination is a rule, it's extremely common.
Your initial isolation procedure was for extracellular particles. What about viruses that don't produce any particles, that are latent within cells? Right. Have you done any work on human cells to detect some of those?
Very good point. The Merkel cell polyoma inducing skin cancer was detected by sequencing mRNA. So when you're looking for latent non-particle producing, then you'd really want to sequence mRNA. And we have looked for cancer-causing viruses sequencing mRNA. I have not found any, but that would be the way to go about it. Now, the problem here is you have to sequence deeper because you also have to resequence all the host mRNAs. But that would be the way to go at those. I mean, very often when during an active infection, if you take a tissue and you homogenize the tissues, there are cells within the tissues. For example, I showed the inclusion bodies, the sort of crystalline sort of accumulation. So you can get a dose by just basically homogenizing the tissue and then passing that through a filter. But for non-particle producing infections, if you want, then mRNA is the way.
Your E-score means?
E-score is the expectation that what you're finding, the alignment occurs by chance. So an expectation score of 1 means that given the size of the databases and the size of the query that you're aligning against it, it would have occurred by chance. So it's sort of like a p-value, but not quite.
Based on the polymerase fidelity? I mean, I'm just wondering how you would distinguish a new species based on sequence or even amino acid sequence.
Because it aligns with a significant E-score to another viral sequence, but it's not the same. So if you resequence HIV, it's gonna be within 10-20% of the other HIVs. Okay. If you sequence a new Picorna viruses, it might only be like 50%, 40% similar at the amino acid level. Okay. The E-score is just sort of a mathematical way of telling you the significance of the alignments. I mean, you can align anything with anything else. Right. It's just not significant unless there is a common history.
So once you define a new species, is there any work done to see if that manifests itself in terms of any clinical meanings, in terms of if that particular virus interacts differently with the host immune system or has a different phenotype when causing illness, if it causes illness at all?
So the initial discovery does, even if we find it in a I think I understood your question, if you find it in the right tissue, when the person is sick or the animal is sick, it still doesn't nail it that it's a pathogen. I mean, you have to go beyond that. You have to do the epidemiology, you have to do the in situ hybridization, the seroconversion. It's much easier to find the virus than to say it's a pathogen.